Out second year botany 2 marks question, which is an examination point of view. Those may asking in previous papers. Let us discuss today. Okay, these are the some of the very important 2 marks questions. First, you have to concentrate here. From this, almost all we hope 7 to 8 questions. We hope from this 20 questions, but it is the previous questions. So many times they were asking in a examination point of view. IP, let us see in all of you, and let us discuss. Okay, number one, first one is from enzymes. That is, what is apoenzyme and what is cofactor. Here from enzyme chapter, what is in enzyme, two parts are there. That in that, one part is a protein part, one part is a non-protein part. Okay, protein is nothing but amino acids, but here which part of the enzyme is protein part, that we call it as a apoenzyme. Enzyme, it consists two parts. Number one is apoenzyme, the second one is cofactor. Apoenzyme means which is protein part, that part we call it as a apoenzyme. Which is non-protein part, that we call it as a cofactor. He may ask, define the apoenzyme and a cofactor. ఓకేనా, ఈ విధంగా అడుగుతుంది. తెలుగులో కూడా అపో ఎంజైమ్ అంటే ఏంటి, కోఫాక్టర్ అంటే ఏంటి అని అడుగుతుంది. కోఫాక్టర్ అంటే ప్రోటీన్ పార్ట్, ఏదైతే ఎంజైమ్లోని ప్రోటీన్ పార్ట్ ఉందో ఆ ప్రోటీన్ పార్ట్నే అపో ఎంజైమ్ అని అంటారు. ఏదైతే నాన్ ప్రోటీన్ పార్ట్ ఉందో, అంటే ప్రోటీన్ పార్ట్ కానటువంటిది, ప్రోటీన్ భాగం కానటువంటిది, దాన్నే మనం ఏమంటామట అంటే cofactor అంటం జరుగుతుంది. దిస్ ఈజ్ 1 2 mark question. The second one, bolting. The second one, bolting. From phytohormones, we are using some phytohormones. When we are using phytohormones, it will be helpful for the elongation of, when the elongation of inflorescence will be occur, at that time there is a chance to the number of the flowers also increases. When the number of the flowers are increases, gradually the fruits also increases. The farmer may get more yield. Are you get my point? Here, when bolting means the elongation of intranode space. Intranode space means a space present between the two nodes, or the elongation of inflorescence, that we call it as a inflorescence, that we call it as a bolting. Bolting means the elongation of intranodal space at inflorescence or in inflorescence, that we call it as a bolting. Very simple. These are the keywords. If these keywords present in your answer, definitely you will get 2 marks out of 2. Okay, the third one, test cross. Test cross also they may ask in so many exams. They may ask when we are hybridization. In that hybridization, we are get first one F1 generation. When we cross between the F1 generation and homozygotic recessive genes, we call it as test cross. Next one, chromosomal theory. Who may propose chromosomal theory? Very simple, Sutton and Boveri. There is a two scientists are there. They propose chromosomal theory. Okay, this also very important. Very simple. These are, if you have in your answer Sutton and Boveri, you get 2 marks. That's it. Next one, phenotypic ratio and genotype. Phenotype and genotype. From genetics, phenotype means we see an organism externally, or external structure of a organism, or external structure of a fruit, which may seen with our naked eye, which may see the external characters, the external shape, external color, that we call it as a phenotype. ఏదైతే మన కళ్ళకి బాహ్య స్వరూపం కనిపిస్తుందో అంటే బయటకి కనిపించేటటువంటి స్వరూపాన్ని ఫీనోటైప్ అంటారు. జీనోటైప్, ఇట్ ఈస్ ఏ మ్యాపింగ్ ఆఫ్ ద జీన్ ఆర్ మ్యాపింగ్ ఆఫ్ ద జీన్స్ ఆర్ జెనెటికల్ మ్యాపింగ్. ఫర్ ఎగ్జాంపుల్, లుక్ హియర్, a gene is there. That gene only, it can be expressed externally, that we call it as a phenotyping. A gene which is responsible for external characters, a gene which is useful for the external characters, that genotype, that mapping, we call it as genotype. Very simple. జన్యు రూపాన్నేమో జీనోటైప్ అంటారు. అంటే జీన్స్ ఎటువంటి జీన్స్ను కలిగి ఉంది అనే అంశాన్ని జీనోటైప్ అంటారు. ఓకేనా? ఏ రకమైనటువంటి ఎక్స్ప్రెషన్ ఉంది అంటే ద జీన్ ఇట్ ఎక్స్ప్రెస్ ఎక్స్టర్నల్లీ, దట్ వీ కాల్ ఇట్ యాజ్ ఏ ఫీనోటైప్. ఓకేనా? దృశ్య రూపం, జన్యు రూపం. ఓకేనా? నెక్స్ట్ వన్, సిక్స్త్ వన్, heterochromatin and euchromatin. The in chromosomes, chromatin is there. For example, here, this is the chromosome. In that chromosome, DNA will be there, that we call it as a chromatin. It is nothing but DNA with proteins, that we call it as a chromatin. That chromatin, it may have, when we are staining, it get more stain, that we call it as a heterochromatin, which may stain in darkly, that we call it as a, because of there is a thick chromatin is there. A thick chromatin, due to the thick chromatin, it leads to the more staining. It is a very compact in nature. It is very contract in nature, or it is a very closer packed. So that we call it as a heterochromatin. It is transcriptionally inactive. Transcriptionally inactive means there is a chromosomes or chromatin is a tightly packed, so there is a somewhat inactiveness is there. Due to it, the transcription is a very inactive. Transcription means, already you people know, transcription means the DNA it synthesis mRNA. In the process of protein synthesis, the DNA which may present in nucleus, that DNA it synthesis mRNA, messenger RNA. That process we call it as transcription. In that transcription, these heterochromatin is inactive. Okay? Next, the opposite to the heterochromatin, euchromatin. Okay? It is a loosely packed and low stained. Okay? Low stained and more active. Okay? Transcriptionally, it is active. Due to why? Due to the loosely packed, or due to the it is a somewhat looser than the heterochromatin. So it is a transcription here. There is a very actively participate in transcription. Heterochromatin and euchromatin, depending upon their thickness, depending upon their closely packed, that these two are divided. Okay, heterochromatin and euchromatin. Next one, components of the DNA nucleotide. Already we are seeing in first year, nucleotide, nucleoside. Whenever the DNA is polynucleotide chain. Polynucleotide chain. This polynucleotide chain may build with three components. Number one, nitrogen base. Nitrogen bases are nothing but guanine, adenosine, cytosine, thymine. Okay, and also it may present in the uracil, it may present in RNA also. Okay, here we may ask you what is nucleotide components? Which components are present in nucleotide? ఓకే, ఏ రకమైన కాంపోనెంట్స్ని కలిగి ఉంటుంది న్యూక్లియోటైడ్ అనేటటువంటిది అంటే వెరీ సింపుల్. హియర్ త్రీ కాంపోనెంట్స్ ఆర్ ప్రజెంట్. నెంబర్ వన్ ఈజ్ నైట్రోజన్ బేసెస్, నత్రజని క్షారాలు. ఓకే, దట్ ఈజ్ నథింగ్ బట్ adenosine, guanosine, cytosine, thymine. Next one, pentose sugar. Pentose sugar, if it is the DNA, that is deoxyribose sugar. If it is the RNA, ribose sugar. Whatever it is, that is the pentose sugar. Next one is the phosphate molecule. Number one, the components of the nucleotide, in the sense DNA components. DNA means polynucleotide chain. That polynucleotide chain means poly means number of. In that number of polynucleotide chain, one nucleotide component. If what about components present in one nucleotide? That is the discuss here. That is number one, nitrogen base, pentose sugar, and phosphate molecule. Next one, eighth question. Transcription unit. Transcription unit. Already we know what is transcription. Transcription means the DNA, its synthesis, mRNA. That process we call it as a transcription. Okay, that process we call it as a transcription. In that, what are the transcription units? Okay, which it means what type of the units present in transcription. In that, number one, protein. Number one, promoter. Number two, structural genes. Number three, terminator genes. Okay, these three units are present in transcription unit. Okay, or we can call promoter genes, structural genes, and terminator genes. These three are present in the transcription unit. Next one, exon and intron. Exons and introns. Children, look here. Very simple. In DNA, some codons are coding a protein or a amino acid, but some codons may not code non-coding region. Non-coding region, it do not code any amino acid. Once again, in DNA sequence, you think it is the DNA. In this DNA, already we know the information present on DNA in codons are according nitrogen bases. Okay, the triplet codons are present in DNA. In this DNA, the total DNA do not have information, do not code protein, do not code amino acids. It do not code, all the DNA may not have coding sequence. Which region or which part are coding sequence, that we call it as exons. Which part may not code any amino acid, that we call it as introns. Very simple. The DNA, it consists, the number of the nucleotides it consists, but all triplet codons or all codons do not code some area. It codes some amino acid, but some region it do not code anything. So which is the coding region that we call it as a exon, which is the non-coding region that we call it as a intron. Next one, capping and tailing. Very simple. These all are belongs to the transcription. Okay. In that, already we see what is transcription. We know from DNA to mRNA, from DNA to DNA to mRNA. When this mRNA comes out, at that time 5 prime end, 3 prime ends, 2 ends are there. 5 prime end, it have to be guanosine triphosphate, methyl guanosine triphosphate. Okay, that we call it as a capping. Very simple. This mRNA, it will be capped. It enclosed with the methyl guanosine triphosphate. It form a cap-like structure, or it form a covering, or it enclosed with methyl guanosine triphosphate. That we call it as a capping. Like that, at 3 prime end, it add nearly 200 to 300 adenosine or adenosines. That we call it as a tail or tailing. We call it as a tailing. Okay. At 3 prime end, addition of the adenosines nearly 300, 200 to 300. That we call it as a tailing. At a 5 prime end, the methyl guanosine triphosphate will be enclosed at 5 prime end. That we call it as a capping at the mRNA. These are the 2 marks questions. So let us see that is stopping codons. Here transcription is going on. Already we seen transcription. This transcription also, when it started and it's stopping, that codons we call it as a stopping codons. Which codon may involve to stop the transcription, that we call it as a stopping codons. What is that? They are UAA, UAG, UGA. Remember, very simple. All are present U and G combinations. U, A and G combination. UAA, UAG, UGA. Look here, my dear friends, very simple. Remember it. U and A and G combination. Okay. Uracil, adenosine, adenosine. Uracil, adenine, adenine, whatever that is. UAA, UAGA and UGA. These are the stopping codons. Stopping codons. Okay. The next one, the 12th question. Point mutation. What is mutation? Mutation is a sudden change may occur in the DNA sequence. Sudden change may occur in genetic material. That we call it as mutation. If that mutations are different types, but in examination point of view, they may ask point mutation. Point means at a specific region or at specific site. Look here, very simple. A single base pair. A single base pair. Okay, DNA sequence are nothing but number of the base pairs are there, but the mutation may occur at single base pair. That we call it as a, or that change, that mutation we call it as a point mutation. Okay, point mutation. Next one. He may ask like this. In the DNA, thiamines are present. thiamine is present at 30%. What about others? Whenever you think DNA, the major component, major nitrogen bases are how many? 4 types are there. Number 1 is adenosine, guanosine, cytosine, thiamine. In that, adenosine always pairs with the thiamine. Guanosine always pairs with the cytosine. Means if adenosine is 30%, thiamine is 30%. What about adenosine? Adenosine also 30%. Means 30 plus 30, 60% is completed. Remaining how many percent are there? 40. In this 40 percentage, 2 remaining nitrogen bases are there, that is guanosine and cytosine. If you share this this 40 percentage to guanosine and cytosine means 20 and 20 percentage. Look here, you have to write very simply. Thiamine, if thiamine, how much percentage is present in thiamine? Definitely the adenosine percentage also that only 30%. The remaining guanosine and cytosine that is 20%. These are the question may ask. Okay, next one. Biotechnology. Define the biotechnology. May ask like this. Biotechnology means we are using technology for the human welfare by using the living organisms. We may apply the which may useful to the human welfare. Some microorganisms are different types of the organisms we are using here, but gene and number of the living organisms we are using here, but we include for that we are include the technology also involving in this, that we call it as a biotechnology. The technology or the organisms we are using for the human welfare, that we call it as a biotechnology. Next one, molecular scissors. Already we see in DNA technology, when we are using scissor, the use of the scissor, it is cut a thing, but here the things are cut. Some enzymes it may cut to the molecules. The enzymes it cut to molecule, that is the DNA, that we call it as a molecular scissors. We are using enzymes for cutting the DNA in DNA technology, that we call it as a molecular scissors. Anukateralu, molecular scissors. Okay, example for that, eco-RI. Okay, molecular scissors means it cut the molecules which we cannot see with our naked eye, that we call it as a molecular scissors. Example for this, eco-RI. Okay, next one, artificial plasmid. Example for artificial plasmid, PBr 322. Example for the artificial plasmid, PBr 322. That is, P means plasmid. Plasmid means already we know the extra chromosomal DNA, which we are using that plasmid as a vector. That vectors, that plasmids are we synthesize artificially. The first artificially they synthesize plasmid, the PBr. B means Bolivar and Redrich. There is a 2 scientists, they synthesize plasmid, that so that in order to honor of them we give PBr 322. Okay, this is an artificial plasmid. Next one, downstream process. Downstream process means in RDNA technology, the last step in RDNA technology, the last step we can collect the product which is preparing, which is processed. At last we collect that product, that we call it as a downstream process. The separation of products in RDNA technology, which product we can obtain, from RDNA, that product we collect and we separate, that we call it as a RDNA technology. In that RDNA technology, that we call it as a downstream process. Next one, cheese. Swiss cheese. The longer amount of the oxygen, the larger amount of the carbon dioxide will become out, that it form a perforate structures, that we call it as a Swiss cheese. The next one is the more The next one is the important enzymes which is using in the biotechnology. That is amylase, lipase, and streptokinase. The important enzymes which are using in industrial. In industrial purpose, we are using different types of the enzymes. In that, the important enzymes are amylase, lipase, streptokinase. These are the enzymes we are using in industrial purpose. Okay, these are the 2 marks questions. Some more. This is the part 1. We can discuss some more. Some more questions in next session. Okay, I hope you all are understood. I think so. But please don't forget subscribe our channel and like and share. Support us. Thank you.